Okay. Hello, everybody. We are back. Look at lecture 7, which is about protein folding. I believe lecture 8 is also. Oh, no, sorry. Lecture 8. Lecture 8 is the ligand binding. I think, yeah. And you guys go into like allostery with hemoglobin and then like other things before you take your exam. So, yeah. Your exam is on October 17th, I believe, for those of you who are watching this in 2020 fall. So, yeah. All right, let's get started. So, protein denaturation, renaturation. Yeah, so these are some interesting things. You guys, honestly, you're not tested on a lot of these. Um, you're tested maybe on this a little bit. Like my exam had some of this, right? It's just a nice overarching general concept to know. Chaperones are also helpful to know, but I don't remember ever being tested on it. It might show up in like a problem set every now and then, but I don't really think it's that important. Just know that they're a thing. But yeah, let's go ahead and keep going. Regarding protein solubility, right? Um, remember that the pi is this midpoint where the charge of a protein is 0, the net charge is 0, and at that point the protein is very insoluble. Anything either side of that, and the lower the pH is, the more positive the protein is. The higher the pH is, the more negative the protein. Okay. And this pi number, right, is dependent on the number of basic residues versus the number of acidic residues. If there's more acidic residues, it's going to be lower than 7, and if there's more basic residues, it's going to be above 7, right? And right, here's just an example of that, right? So right now, if you right, so at the protein isolation point, presence of both positive and negative charge, right, which makes it 0, which makes it insoluble, right? Um, this is just a nice image kind of showing that here, right? At the pi, the net charge is 0, right? Promoting aggregation and precipitation of the protein, right? When it's above or below its isoelectric point, right? In this case, it's technically above, right? The isoelectric point is because it's negatively charged, right? They repel each other and they will not precipitate out. Just this idea that we can use pH. You can use pH to predict the pi, right? So like if you change the pH, right, and the solubility of the protein decreases like crazy, you know that the area that it decreased around like decreased a lot, and like it's very little around it. Basically, the area where there's a lot of precipitate forming is the pi, right? So there's a lot of precipitate here for protein A, a lot of precipitate here for protein B. Those must be the pi's at around 4.2 ish, maybe, and like 8, right? um, Talked about salting out, right? This idea that if you add enough salt, depending on the protein, right, you'll eventually cause the protein to aggregate together and precipitate out, right? Chromatography, right? Different column chromatography, affinity size exclusion, chromatography, ion exchange, right? Just different techniques for getting proteins and analyzing them, right? And this A280 thing is specifically asking about tyrosine and tryptophan. They do ask about this on the exam, specifically which amino acids absorb this A280, the absorbance 280 nanometers. It's tyrosine and tryptophan. So just be aware of that if they ask, right? Yep, so protein purity, right? We can look at SDS page, right? You load these protein samples, you apply a current from negative to positive, right? You analyze the gel, right? But yeah, other than that, we can get on to the actual lecture. The cool thing, David Baker, UW PI, pretty cool guy. I actually took a class with his wife, Rahola Baker, but yeah, Hanala was great. Hanala Rahola Baker, she's pretty cool. The class was pretty difficult, but a lot of experimental technique. But what do you expect of the wife of somebody who just won the Nobel Prize? So that was a really fun class. I really enjoyed it. So yeah, AlphaFold, Rosetta Fold, not important for the exam. Just cool different protein modeling things that show you how proteins fold. Very interesting models, by the way. Like proteins are so cool. Yeah, okay, here we go. So basically, right, the primary sequence, right, encodes the amino acids, which encode the structure, right, because the amino acids are what decide the structure, right? The folding process must be spontaneous, so negative delta G, right? Otherwise, the folding would never happen, right? And it needs to be a structured balance between these unstructured regions and structured states, right? So we have to go from this blob to this organized thing of alpha helices and beta sheets, right? So the folding is driven by the increasing entropy of solvent and water, and also the release of heat from forming these intramolecular interactions. So like if you form H bonds, ionic interactions, covalent bonds, right, you actually release heat when you do them because those are favorable to do, right? Bonding is favorable, generally speaking. In this case, it's also entropically favorable, right? Because just remember that hydrophobic, a lot of the proteins have like hydrophobic insides, right? So if you expose those hydrophobic insides to the outside of the water, outside to the water, right, the water will try and cover it up. And that kind of forces the water into these certain states. That's not very disordered. It's very organized, right? The water has to be stuck in these certain positions to keep the hydrophobic regions from disrupting the water, the rest of the water molecules, right? However, if the protein is very nicely folded and the hydrophobic stuff is on the inside, the water is free to do whatever the heck it wants, and it can run around the protein however it feels, right? But yeah, this idea that this is like a slide, right, is basically saying that like it doesn't just magically fold in one go, right? It takes a bunch of tries, right? It'll fold in one way and then realize that it's not good enough, and then it'll refold into a different way and then realize it's not good enough. And eventually, after some time, it'll eventually fold into the correct conformation, right? After folding over and over and over and over again, right? Until it reaches that position where it can no longer fold anymore because it's in its stable state and it's happy in that stable state. Uh, Proteostasis is just a fancy way of, by the way, of saying like protein stasis. I believe it's just like a way of saying that it's in its happy form. So they must fold into their native structure, right? Start at the ribosome, right, as a primary structure. Fold into its secondary structures, right, and then fold into its full tertiary structure, right, when it combines these secondary structures. And if it doesn't fold properly, right, it can use chaperone proteins to help fold correctly. And yeah, unfolding is the reverse reaction, right? But yeah, you can't go from a folded protein back into the ribosome, right? But you can go from a folded protein back into the primary structure. You have to disrupt these hydrogen bonds that hold these secondary structures together, right? I talked about before in one of these lectures how the main thing that holds secondary structures together are hydrogen bonds, right? So just be aware of that. Yeah. There's a lot of stuff going on this slide. All this to say that, sometimes when an organism is stressed or your cells are stressed, right, it can cause mutations, right, in these proteins, right, and that leads to many different things like aggregation, which leads to a disease. So Alzheimer's is actually an example of that, as well as Parkinson's, I believe, with Lewy bodies. But Alzheimer's, like amyloid beta plaques, I'm sure you've heard of them before. The way they actually form is it's actually a protein that is basically aggregating. It's misfolding and then aggregating, if I'm not mistaken. Um, and that aggregation is linked to Alzheimer's, but it's not the cause of Alzheimer's. It has actually been proven that the amyloid beta plaques are not the cause of Alzheimer's, because if you actually remove the amyloid beta plaques after someone has gotten Alzheimer's doesn't just disappear. So the amyloid beta plaques are thought to actually be a byproduct of whatever is actually causing Alzheimer's. Yeah, so we've got different folding properties, right? We've got here folded, native, right? Is these the same thing, or unfolded / denatured, right? And you can unfold proteins by using things like heat. You can use beta mercapta ethanol, right, for chemical denaturants, and you can also use SDS. So this is temperature, right? This is SDS, and this is beta mercapta ethanol. I'm just gonna say beta, right? So either SDS or beta mercapta ethanol, right, for chemical denaturants, right? And you can measure the property. You can measure how folding occurs based off of fluorescence, right? You can add fluorescent tags, right, so that fluoresce when the protein starts to unfold, stuff like that. You can use all these. I can't remember what CD stands for. It's some sort of rotation, and I can't remember. It's like a rotating plane polarized light, if I'm not mistaken. I can't quite remember off the top of my head, but it shouldn't be that important for this class. And here's like the fraction, like TM, right? The fraction of when 50% of the protein is unfolded. I've never had to use this equation before personally. It's probably useful to know, but I would just keep it in the back of your mind. What is important to know is what TM means, and TM is just the point at which the temperature at which the protein is half folded, half unfolded, right? Same thing with this denaturant 50%, the concentration of denaturant at which the protein is one half folded, right? And the idea is that if you have a higher TM, that means it takes longer, or like a higher TM or a higher denaturant 50%. It takes more temperature or more concentration of denaturant, right, to actually destabilize the protein. Like you can see that here, right? For example, right, this protein has a 50% unfolded rate, right, at around, I don't know, like 55-ish, right? But this one is around 65, 70 maybe, right? So the TM is very important, right? If you look at this, right, this protein here doesn't unfold till around, like, I want to say 3.4-ish concentration of guanidinium chloride, right? So this is something I actually did talk about in my exam review as well for exam 1. There's a question about this, so just keep that in mind. Okay, so this is an important thing to keep in mind. So disulfide bonds are actually really important because if you denature and disrupt the disulfide bonds by reducing them, you have to make sure that if you want the native structure back, you do things in the correct order. So, let's take a look at this. If you see here, let me try and see what I want to talk about exactly. So, the idea here, right, is that you add urea to denature your protein. You also add beta mercaptoethanol, right, to reduce these disulfide bonds, right? So basically what you're doing is first you reduce the disulfide bonds, right, and you break them all apart, right? Right. And the other thing you do is that you add this urea, and the urea disrupts any secondary interactions. So like any hydrogen bondings, like maybe between amino acids here or something, those get broken up as well, and it starts to stretch out this protein into like its primary structure. It starts to look a little bit more like this maybe, right? It becomes more and more like linear basically. It's no longer in this tertiary 3D shape basically. But yeah, basically, if you remove the urea first, right? Like you have this structure, right, where it's like all the disulfide bonds are gone and those secondary interactions have been removed, right? If you remove the urea first, the protein is able to refold, and then if you remove the beta mercaptoethanol, those disulfide bonds will reform where they were initially because the protein has been given the opportunity to refold into the position it's supposed to. If you remove the beta mercaptoethanol first before the urea, the disulfide bonds will form, right, in places where they're not supposed to. And even if you remove the urea, because you've formed covalent bonds now, right, the protein is now stuck in the state that you put it at before, right? So this is kind of reiterating it. This RNase is a specific protein, right? Has 4 disulfide bonds, right, with unique pairings. If you unfold it with urea and that, right, allow refolding, right? You allow refolding, right? Remove the urea first. You allow, and then you allow the disulfide bonds to form after removing beta mercaptoethanol, right? That's what I said. If you remove beta mercaptoethanol first, right, you completely destroy the protein's function, as the protein is no longer able to reform into its normal structure before it's oxidized into the covalently bonded disulfide state, right? And then, like I said here, many possible wrong pairs of cysteines, right? Only the correctly folded structure puts cysteine pairs together, right? So you must remove the denaturant SDS, or urea in this case, before oxidizing and removing the beta mercaptoethanol. This is kind of just talking about what I talked about before. I don't think this is too important. You should know, alpha helices and beta turns, hydrophobic segregation. Hydrophobic things will be clumped together, and hydrophilic things will be clumped together, right? But yeah, I'm trying to think if there is like there's like a thing I want to talk about. I guess the main thing here is that the idea that certain amino acid sequences, like that I+3, I+4, and that polar, nonpolar kind of thing for the beta sheets, and the alpha helicities for that I+3,4 thing. Those structures actually form first, right? So you see how this beta sheet is forming, this alpha helix is forming, right? This beta sheet is forming, right? And then these, they all basically come together. They form first, and then they come together, right, to form the tertiary structure of the protein, right? So folding is not it's not random, right? There's some order to it, right? Like these sections are turned into this structure, right, because of the amino acid sequence, right? These sections are turned into this structure because of the amino acid sequence, right? Things like that. But yeah, the structure of the tertiary structure of the protein is a little bit random, I would say. I mean, it takes some time, right? It has to randomly try different conformations before it can actually find a proper conformation, right? But like the formation of these secondary structures is pretty um, is pretty like one directional. I would say, I guess. Um, I don't think this matters pretty much at all. Like this is cool. Like yeah, it's great that this house is a crowded place and you have a lot of stuff, but I don't think it matters. Um, yeah, so this is the idea that some proteins need refolding help, right? So you can imagine, right, that mistakes happen, right? Even cells make mistakes, right? And so eventually, if you make four proteins, right, one of them might become fine, right? But the other three, might be bad, right? They might be toxic, right? They might be misfolded, right? So how do we get rid of these proteins or fold them again so they're actually in the correct state? Well, that's where we get to help proteins, such as the heat shock proteins, right? These are the cell first responders, right? And they're induced by these heat shock proteins are induced by, guessed it, heat shock, right? So heat or oxidative stress or acidosis or chemotherapy. Right? These are things that cause mutations in maybe DNA or mutations in proteins, right? And or they just denature the protein itself and cause it to be inactive or non-functional or maybe even toxic to the body, right? Um, so this heat shock factor actually, it upregulates um transcription is basically upregulated by these heat shock factors. Um, but yeah, and it basically binds to partly folded proteins and prevents formation of these aggregates, right? It's trying to prevent these aggregate formations. And it's also ATP independent, which is really cool. So basically this protein is just like it's a protein that binds to proteins that have not formed correctly and stops them from aggregating and forming these insoluble clumps, which can cause severe problems. There's other types of heat shock proteins. There's all these things, but I don't think these are too important. Just know that these things exist, right? And that they basically help with folding proteins or stopping them from aggregating, right? See how there's like yeah, see here's like another one, right? So HSP 40, 70, ATP, right? So ATP required for this one here, right? Oldase, right? Different enzymes. These are different, protects hydrophobics, right? These are different enzymes used to refold proteins or stop them from aggregating, right? But yeah, concept, right? So if they bind to exposed hydrophobic regions, remember I talked about before, hydrophobic regions, right, cause water to try and cover up those hydrophobic regions, and we end up with a decrease in entropy, which is bad. The universe does not like a decrease in entropy, right? This these hydrophobic regions being exposed suggests a misfolding of proteins because proteins normally do not put hydrophobic regions in the hydrophilic water, right? So properties of heat shock, right? It has to be very quick to bind to proteins, right? It has to bind to hydrophobic surfaces and it has to be non-specific. You might wonder, why is it non-specific? Well, remember, there are millions of proteins in the body, right? You need to be able to have your heat shock proteins bind to all of them, right? If it only binds to one type of protein, then you've got a problem, because what if the other proteins misfold? You need the heat shock protein to be able to bind to a lot of different proteins. So we need the protein to be non-specific so that it can bind to any protein available in the body that misfolds so that it can actually refold it correctly. And yeah, now you have chaperones. These chaperones are the ones that actually fix proteins. So they basically take misfolded proteins, right? They put it inside this little barrel, right? They do a little shake, shimmy, 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 right? Using ATP, right? And then they fold the protein, right? And they come out, right? And then the protein is folded, right? Generally speaking. This is an example of grow ES, grow EL. You might get a question or two on this in the packets, but I've never been tested on this. But it's good to keep in mind and to know how this mechanism works. Is there anything else I want to talk about here? I guess I'll talk about this briefly. So for the grow EL, grow ES complex, right? Basically, you have this grow EL subunit, and then you put a protein in, you cap it with the lid called grow ES, right? And you bind this ATP to it, right? You then release the 7 ADPs, and you cause ATP hydrolysis, right? Which leads to the folding of the protein, right? And then you bind ATP to the bottom part of the grow EL, and then you hydrolyze that, right? And then you basically redo this process over and over again. But yeah, is there anything else? Yeah. Also, to keep just remember that like here I forgot to mention that over here, right, the cap is removed and the protein is the refolded protein is removed. Basically, this ATP hydrolysis, right, is what actually causes the protein to actually be refolded again, right? And there's basically two sides to this enzyme. So one side, there's two grow ESs, one binds to the top, one binds to the bottom, right? And there's two compartments for two different proteins to refold, right? Basically, when one refolds, the other is basically being put in, and then when the other one of them, the refolded protein is released, the other one is being turned into its full. Again, like I said, this is not too important. If you want to go over this in more detail, feel free, but generally I have not seen this on an exam, so keep that in mind. Chaperonins, multi-subunit complex. Yeah, again, multi-subunit. There's many different subunits, right? And you can, this is a side view of how it looks, right? But yeah. So I would say, yeah, so the entry vessel, right? So the way you enter these is that there must be hydrophobic regions that are exposed, which is how the chaperones are actually able to bind the proteins in the first place. And you would think that like the inside of these proteins is probably going to be hydrophobic, right? Maybe hydrophilic, right? And probably a bit of both. You know, there are probably going to be some hydrophobic things to bind to these proteins to actually help them fold. And it might be some hydrophilics to help make some bonds, some sort of ionic interactions or hydrogen bonds with the proteins, right? That are being put inside of this thing. So, that in mind. And here is a list of protein diseases, of protein misfolding diseases. I mentioned Alzheimer's, and I believe I mentioned Parkinson's with Lewy bodies. So, yep. Here's amyloid, right? Like I talked about with Alzheimer's, right? Yeah, this is not really that important. These are cool. I would recommend looking this up if you're interested in these. Like prion diseases are pretty interesting to look at, but I wouldn't be too. But yeah, yeah, melting curves, the whole idea of TM, right? Yeah, functions of three types of chaperones. So like heat shock proteins, the grow ES, grow EL, like how they work basically. And yeah, I guess I get these are I don't know. I feel like these are a little bit specific. You don't really have to know. I guess you just for amyloid just know it's a misfolded aggregation of protein. The aggregation of misfolded proteins, right? And what are its properties? They're proteins that don't work and they are insoluble, right? Because they aggregate. Right? I guess I'm trying to figure out for the chaperones. I think the like, heat shock proteins are important to realize. In that I don't think that important. I think that's really all. I think there's probably like where is it? This whole idea of refolding, right? This will most definitely show up on the exam. Like it showed up on my test, this idea of the order of things that you put in and take out, right? Just remember if you want your protein to refold correctly, right? You need to first, if you put both denaturing agents like SDS like in there and a reducing agent, you need to remove the denaturing agent first before you remove the reducing agent. because you want the protein to refold into its normal its normal structure, normal construction, right? Before you take out the reducing agent and oxidize the bonds together, right? But other than that, I hope this video was helpful. Definitely a very short one. I feel like I'm making these videos shorter and shorter. But hopefully they're at least somewhat helpful. I'm trying to make sure that I'm not covering like everything in these slides because I don't think everything is important. But hopefully this is enough to at least study off of, and you could get an idea of what the exams are looking for. But yeah, have a good one, guys, and thank you for watching.